Hello, students. I am Obasi. I am Mrs. Obasi, IP, your biology teacher. The last time I was here, we started discussing this genetics, the biology of hereditary, and I told you that by the time I come back, we would continue. If you can remember from the last time we discussed, we talked about the various words they are supposed to know in genetics, words that keeps reoccurring. They are supposed to know their definitions, what they mean, and where they fit in. We also discussed DNA. We discussed about the helix structure of the DNA and the chemical components of the DNA and its structure. So today I'm supposed to continue with the other part of the lesson. But by the end of this particular lesson, you should be able to state the functions of the DNA, 1, 2, explain segregation of genes at meiosis, 3, you are also supposed to explain probability in genetics, and then state the principles of probability. So let us start first with the functions of the DNA. Okay, the first function of DNA here that I want to talk about is that the DNA keeps the number of chromosomes peculiar to an organism constant from generation to generation. And it does this by the doubling or the duplication of the cells during the process of meiosis and mitosis, which you are already familiar with. This newly developed cells now contains the complementary number of the chromosomes of the parents. Okay? Then the second function of DNA is that it controls growth and development of every living thing by controlling the protein and enzyme synthesis. Okay? Now when we're talking about protein synthesis, what comes to our mind is growth. Protein synthesis has to do with growth, protein has to do with growth, and enzymes have to do with development. So if we say that the DNA controls the growth and development of every living thing and then it's by controlling the protein and enzyme synthesis, that means that without the DNA, there wouldn't be protein and enzyme synthesis. And without protein and enzyme synthesis, there would not be growth and development. So the question now is that how does the DNA control the protein and enzyme synthesis? It does this by using 3 types of RNA. Already the last time I was in class, we talked about RNA, what it means. We also talked about DNA. I told you the differences, how we can differentiate RNA from DNA, and what is inside them. So today, as we are talking about DNA, we are talking about the uses and types of RNA that the DNA uses to ensure protein and enzyme synthesis. The first one is what we call the messenger RNA. We write it in this way, messenger RNA. Okay? This messenger RNA is dispatched into the cytoplasm. Remember when we talked the last time, we discussed where we find the nucleus. I told you that you find the DNA in the chromosome, and the chromosome is inside the nucleus. The nucleus is inside the cell, and surrounding the nucleus is the cytoplasm. Okay? Now, if you remember from the last time too, I told you that the nucleus is responsible for writing command that is transferred to the cytoplasm, which bears the other organelles inside the cell. It is not the cytoplasm that will ensure that this command that has been given is carried out. Okay? Now, if you can still remember that, you will now understand what I mean when I say that the DNA releases the messenger RNA into the cytoplasm. Okay? Now, the function of this messenger RNA is that it is responsible for specific codes that are dispatched for protein synthesis. Now, when we are discussing protein synthesis, I told you before, we are discussing about growth. Now, there are codes that are dispatched by the DNA for protein synthesis. These codes are dispatched through the messenger RNA. Okay? So when the DNA releases the messenger RNA into the cytoplasm, it dispatches codes to continue with the process of protein synthesis. The second way it does it is through the use of the ribosomal RNA. ribosomal RNA. We write it like this, ribosomal RNA. Now, when we hear about the ribosome as a structure in the cell, what comes to our mind is there is a structure for protein synthesis. It helps in synthesizing protein. That's what this structure does. Okay? Now, what happens here is that the DNA controls the synthesis of ribosomal RNA. Okay? And later dispatches it to cytoplasm, where it is responsible for attachment of the ribosomes on the messenger RNA. Okay? Remember I told you that the messenger RNA is responsible for dispatching codes for protein synthesis. Well, the ribosomal RNA is dispatched into the cytoplasm so that it can help to attach the ribosomes to the messenger RNA. We are still talking about this same protein synthesis. Then the third one here we are going to talk about is the transfer RNA, which is written like this, tRNA. Okay? The transfer RNA is the third RNA that the DNA uses to ensure protein and enzyme synthesis. Okay? What the DNA does is that it dispatches the transfer RNA into the cytoplasm, where it picks and hands over various amino acids to the ribosome to be joined together in long chain during protein synthesis. Now, if you can think back, remember that what amino acids are. Alright? The amino acids are the final substances that are obtained after the breakdown of protein in the body. Also, even though they are the minutest form of this protein that you can see, they also serve as a building block for protein synthesis. So when this transfer RNA is dispatched into the cytoplasm, it picks up various amino acids of different forms, and when it picks them up, it goes and joins them with the ribosomes. And the ribosomes are responsible for protein synthesis. So it helps ribosome to build up the protein. By the time it hands it over to the ribosome now uses it to create a long protein chain. Okay? So the DNA actually helps in the formation of, protein and enzyme synthesis, which helps in growth and development. But it does it with the help of the RNA. Alright? So if you can refer back to the last lesson, you remember where I discussed what RNA is in earnest and the differences between DNA and RNA. Okay? So that's the first thing. We've discussed the functions of the DNA. So the next thing I want us to discuss is the segregation of genes at meiosis. Okay? The next thing I want us to discuss is the segregation of genes at meiosis. Excuse me. Okay? We'll discuss the segregation of genes at meiosis. Okay? We already know that meiosis, which you have already learned about, produces haploid gametes. Alright? If you can remember vividly the last time we discussed about diploid and haploid gametes. We know that the diploid number for human beings is 46. The haploid number is 43. We also know that when we are discussing haploid number, we are discussing the gametes. The gametes of living organisms or of humans have that haploid number of 23. Okay? So meiosis produces haploid gametes. Two genes segregate at the pair of homologous chromosomes. Now, when we are discussing meiosis generally or gene duplication, we are talking about chromosomes, and these chromosomes come in pair. We call this pair homologous chromosomes. They come in pair that we call homologous chromosomes. If you can remember very vividly. Okay? Every one of these chromosomes carry what we call an allele. Okay? When we are discussing allele, we are talking about specific genes, variants of specific genes. When you talk about a particular allele, it carries a particular character. For instance, if we are talking about the height of a human being, okay? If you are talking about height, if the character we're trying to talk about is the height, the allele is what carries the variants. You know, when you're talking about height, you have tall and short. Alright? If you're talking about eye color, you might have brown, and black. If you're talking about hair color in humans or in blacks, we have brown and black. So, okay. So this allele when we are discussing alleles, okay, we are now discussing the genes that carry these specific characteristics. So when you say the allele is has variants of a particular gene, it means that we are talking about a particular character that have different variants. Now, a particular location where these things are located on the chromosome is what we call the locus, as we discussed earlier. Okay? Now, let us say that the variant for tallness is A, capital A. Usually, if you have it like that, that means the variant for shortness will be small letter A. Alright? If you say the variant for black hair color is capital D, the variant for for brown hair color might be capital D, small letter D. But it means that we are talking about the same character, but the way it is shown in different ways. So, here, the allele, when during cell division or during mitosis, these chromosomes divide. They come out separately as single chromosomes. Remember, they are pairs of homologous chromosomes. They come out as single chromosomes. So if we have, maybe have A here, we have A here, we have B here, we have B here. Okay? They have come out as simple chromosomes. So during gene combination, finally, one combines with the other. You understand? It doesn't combine in a specific way. It combines differently, one with the other, in different ways, intermittently. And when there is combination, that's how we get genetic variations. Alright? Because these chromosomes these are haploid chromosomes. Combining together invariably, we get genetic variations. But if there was no form of variation, we're going to get exactly what the parents have. In other words, we'll be having photocopy of children being photocopies of their parents. Do you understand me? But because of this variation, that's where we can see a child. Your father is black and you are slightly brown. Maybe your father is black and your mother is brown. You are, brown, or between black and brown. So that's what we mean when we are talking about the alleles and the variants that are involved. Okay? So I said here that an allele is a variant form of a given gene. That is one of the two or more versions of a particular gene. Okay? So the next thing I want to talk about, so what we have talked about here now is segregation of genes in mitosis. In mitosis, sorry, I wanted to write mitosis, where the genes separate. Okay? And after the variations and the crossing over, we now get genetic variation in children at the end of everything. Then, another one is that at the end, we have recombination of genes at fertilization. Okay? We already know as students that fertilization is the fusion of the male and female gametes bring out offspring. Okay? Now, it is after this process of crossing over that at fertilization, we now find out that these haploid gametes of the male and female will now fuse together randomly, okay, to produce a zygote. And it's the random nature of the combination during fertilization that contributes to genetic variations of offspring. All right? Then the next thing I want to talk about finally is probability in genetics. The next thing I want to talk about is probability in genetics. Now, generally, what is probability? What is probability? Okay? Probability is the likelihood or chance of an event occurring. It is the likelihood or chance of a particular event occurring. Okay? For instance, you can say the probability of a newborn baby being male or female, okay, is 50%. Alright? The probability of a newborn baby being male or female is 50%. Okay? The probability of throwing a dice and getting one, you understand, is 0.17%. Probability of throwing a dice and getting one, you understand, is 0.17%. Okay, so in genetics, probability can help us to predict the outcome or likelihood of producing a desired character. It can help us to predict the outcome or likelihood of producing a desired character in crops and livestock. Okay? It also gives us also the chance of a child inheriting a genetic disorder from parents can also be determined with a probability. Okay? Now, these probabilities we are talking about, there are three principles of probability. We have three principles of probability. Okay? We have three principles of probability. The first principle is that we say here that the result of one trial of a chance event does not affect the result of the other trial of the same event. The result of one trial of a chance event does not affect the result of the other trials of the same event. Okay? For example, when you are tossing a coin, okay? If you toss a coin now, you can get one. The next one is not a must that because you got one, the first one, you will get two. You know? And because you got two, it doesn't mean you will get three. So, what we're saying here, the result of one trial of a chance event does not affect the result of other trials of that particular event, of the same event, okay? That is the first principle of genetics. We have a second principle. Okay? The second principle here says that the chance that two independent events will occur together, that is at the same time, is a product of their chances of one occurring separately. That is, if one fruit in every four, example now, I'm giving an example, if one fruit in every four in a farm is ripe, and one in every five is sweet, the probability that the fruit you pluck will be ripe and sweet is, what we mean here now is, excuse me, can I clean this? Hope you have gotten this. Okay. Here we are saying that one in a farm now, we say that one fruit in every four in a farm is ripe. Okay? One fruit is ripe. One out of four in every farm is ripe. And then one out of five in every farm is sweet. Okay? Now the probability that the fruit you pluck will be ripe and sweet is going to be, the probability now is going to be 1/4 times 1/5, giving you 1/20. Now look at what I did here and what I explained there. I said that the chance that two independent events will occur together is the product of their chances of occurring separately. Okay? The chance that two independent events will occur together is the product of their chance of occurring differently. Remember, we are talking about product, we are saying multiplication. Okay? So the chance that the fruit you pluck in the farm will be ripe and it will also be sweet is the product of the number of times you have seen the ripe fruit and the sweet one, giving you 1/20. Now, let's talk about the third and the last principle of probability. The third principle of probability Okay. So what I said here, the third principle of probability says, if a particular event can occur in several mutually exclusive ways, if a particular event can occur in several mutually exclusive ways, the probability of the event occurring is the sum of the different ways. Okay? Now, what it's trying to explain, let me use an example to explain it. Let us say Okechukwu walks into this office. Okay? And Okechukwu walks in here dragging his feet. Okay? Let's say he walks in here dragging his feet twice out of a whole day. Alright? Then, maybe after he has been cautioned, he now comes in, walking, raising his feet from the ground. He comes in 3 times. Alright? What we are now trying to say is that the probability of Okechukwu walking in, either dragging his feet or walking straight, or the probability of Okechukwu walking in straight is the sum of the number of times he walked in straight and the number of times he walked in dragging his feet. In other words, the probability of Okechukwu walking in without dragging his feet is 5. Okay? So that is the much we can take from the lesson today. And I want to give us our assignment. So the assignments are, number 1, state 2 functions of the DNA. Number 2, explain how DNA controls protein and enzyme synthesis. Number 3, what is probability? And number 4, state the principles of probability. I hope you have gotten all the assignments. Thank you for listening to me today. I remain Mrs. Obasi IP, your biology teacher. Stay safe.